And then I want to bring it in here, quiz 1. So quiz 1 is here. So the first question is, which of the following experimental organisms would be the most useful for studying birth defects in the development of the spinal cord? So correct answer will be here, zebrafish. So closest to the human. And number 2, transcription, which is synthesis of mRNA from DNA, occurs in what compartment in eukaryotic cells? So obviously, answer will be nucleus answer. And number 3, in a typical eukaryotic cell, which molecule provide most of the direct, that's the key word, chemical energy for performing cellular work, so ATP. And then number 4, which of the following statement is not accurate with respect to prokaryotic cells? And I think there is a little bit of a tweaking here, and in that the yeast is a eukaryotic system. So answer will be B. And then D, how to make a 0.2% NaCl in 10 ml. I think now you are very familiar with this kind of question now. So you're writing 0.2 divided by 100 ml times 10 ml, right? So then you can cancel all units except gram. So it should be something like 0.2 divided by 100 times 10. So that's your conclusion. So you should be able to do that. By the way, when you have exam 1, I think September 16 to 17 or 18, something like that, it'll be 2 days window. I think Dr. Duran will be telling you about this more detail. And you will be required to have your calculator. And so we have to work a little bit with the lockdown browser so that using calculator would be not a inappropriate behavior. So quiz 1 was covered, and we have quiz 2. Let me quickly go over quiz 2. Make it a little bit bigger. Number 1, carbon can form 4 covalent bonds, right? And what is the typical pH of eukaryotic cell? 7.2. What type of chemical bond holds together water molecule? It should be hydrogen bond. And which order of protein structure would be best exemplified by alpha helix and beta sheets secondary? And then we have 5 number, what type of mechanism that regulates protein function involves the release of catalytic subunits for the enzyme to transition from an inactive to an active state via cooperative binding of a ligand. So it should be allostatic transition. And then next question will be like this. You want to make a 50 ml of 0.1 mole NaCl. How many grams of NaCl would you need to make it? So molecular weight is provided here. So it's very straightforward that 58.4 gram times 0.1 times, so you got to convert 50 ml into liter. So 0.05 liter. So you multiply that, okay? One more time, 58 times 0.1 times 0.05, which will be resulting in specific number. That is your answer. So far, if you have any questions, please let me know. So quiz 1 and 2 are covered. Okay, guys, so from now on, if you don't have a specific question, I'm gonna start talking about lab 3. So I'm gonna make it bigger. So, okay. Okay, today lab is lab 3, basically covering data analysis on cell proliferation. Okay, so reminder is this, quiz 3, but I already told you guys that somehow we have lockdown browser problem. So we need to fix this first. So you can try, and I don't know whether it's gonna work or not for you, but in general, I don't think it'll work. So I'll let you know, I'll keep you posted so that you will know what you have to do with this quiz 3. Okay, so I'm making pen here. Choose MSU color. Okay. So what is our next topic? Obviously, one of the most important things that I have to talk about here, class objectives. Number one, I think last class period, all of you had hands-on experience on detaching cells from a flask, and we counted cells, and then we were ready to applying these isolated cell into a 96-well plate. So the second day, you were not participated in, but TA and I prepared, and basically we treated 2 different chemicals here. One of the chemical that we treated to the cancer cell was cisplatin, and the second chemical we treated was PT-DECA2. So these are actually in your probably lab handout, it's labeled as toxin, but actually more accurately, I want to say this as chemotherapeutic agents. Particularly in front of a chemotherapeutic, I would say that cancer, there are chemotherapeutic agents. Okay, so we'll study about general chemical feature of cisplatin and PT-DECA2. What are the similarities in terms of their structure? And what would be potential effects on cancer cell? That's one of the main objectives. And then second, since they are structurally related, right, between them, so you can somehow setting up a working hypothesis on the effect of particularly PT-DECA2 chemicals on cancer cells. And then number 3, in order to see the effects of cisplatin and platinum-DECA2 on cancer cells, we actually do some chemical assay. The assay name is XTT proliferation/viability assay. That will give you actually quantitative results, the data. So we are able to process these quantitative data using standard data analysis method. And eventually we're gonna use Excel, which is well-known program that you guys probably have used before. And Prism 8, this one you probably never used before, right? Using these 2 software, eventually we will perform statistical analysis. Those are our objectives. Let's talk about chemicals that we have used for the class of pure cancer cell. So first of all, this is cisplatin. You have a platinum inside, and then that is associated with the chloride ions, and then amine group. So 2 amines and 2 chlorides. So that's a cisplatin structure. This was developed in 1978. And then since then, it has been used as anti-cancer chemotherapeutic agent. Right? Many types of cancers have been treated with this chemical and quite successful, even though it has a little bit of a side effects with the normal cells. So how this cisplatin works is basically as shown in this category here. Basically, it will be interacting with a purine base of DNA. So purines are actually, examples are A, adenine, and guanine, right? So these 2 guys are target for the association of this cisplatin. If cisplatin is associated with the DNA, it causes a lot of problems, including DNA repair mechanism and DNA transcription and DNA replication. These are all associated with this cisplatin problems. And if DNA get damaged because of this cisplatin, and then cells are dying, as a result, cancer cells are undergoing cell death, which another name is apoptosis. Not only cancer cells are dying with this cisplatin, normal cells also are subjected to die if cisplatin is available, right? Because any rapidly growing cells, like your hairs, like your, some skin cells, particularly your hair, for example, really grow fast. And now those hair cells are target for this cisplatin. So during chemotherapeutic period, cancer patient losing these hairs because they are target too. There are a lot of references on their basics of the action mode of cisplatin, right? And the molecular mechanisms of actions, and reference on cytotoxic effects of cisplatin. So when you write your lab report, you could use these as a reference, right? Report. So you don't need to really reinvent the wheel, but look at this PowerPoint and use these as a citation. Okay, how about platinum deco? So this is PT deco 2. Well, this is very short abbreviated name. The full name is like this: 2-cyano-2 oxamino N prime diethyl amino acetamide. So the structure is this: you have a platinum at the center, and then you have oxygen, nitrogen, and here all connected. Nitrogen again, you have here all carbon chain here, right there. So this is a little bit bigger than cisplatin, but it's similar in that it has platinum in it, right? So platinum is found to be playing a key role that causes interaction with the DNA. So now let's see that. This is short summary about PT deco 2. Number one, it is platinum-derived cyanamixate. And then this has been recently developed by Dr. G. I think I talk about this from chemistry, MSU. I think his full name is Dr. Gerasimchik. So you probably go Google in MSU, find out him from website. And we expect that this could have a high toxicity because the structure of PT deco is similar to that of cisplatin. A little bit of problem is that compared to cisplatin that has 2 chloride, 2 amine, this has, this also has oxygen here, relatively soluble, but some area here has a lot of hydrogens. So that's relatively saturated. So it may be less soluble than cisplatin, right? So reference on PT deco synthesis, you should be able to find in this paper. You could use this as reference for your report. But there is no other reference on its effect on cell viability on cancer cells. That's why we use this for our experiments in our cellular biology course. Okay, so from now on, we'll start to thinking about or setting up hypotheses for the potential effects of platinum deco 2. I gave you already hints that PT deco 2 are structurally related, right, to cisplatin. Structurally related. So you might have, end up come up with one potential idea or hypothesis. 2 possible hypotheses can be set up. One hypothesis, I would say null hypothesis. You know what I mean by null hypothesis, right? I would say the null hypothesis would be that, well, PT, that even though they are structurally related, right? Even though you may end up setting up like, okay, PT deco 2 will have no effect on cancer cell, right? That's one kind of a null hypothesis. That could be one of them, right? Or you could probably end up with a little bit different kind of a hypothesis. For example, you could say that PT-DECA2 effects on cancer cell will be less than that of cisplatin. It could be that, right? But in general, null means no difference. So PT-DECA will have no effects on cancer cell. That will be null hypothesis. And then we could have an alternative hypothesis. Alternative means, well, null maybe is not correct. Then you have to have some kind of alternative hypothesis. This could be that PT-DECA2 effects on cancer cell, right, will be almost equivalent to those by cisplatin. That could be a kind of a alternative. So you see the difference between null and alternative, right? So we'll get this kind of null hypothesis and alternative hypothesis one more time. So you'll see that. Okay, so I have to review a little bit about our experiment, what we have done so far. So background on the experiment. Day 1, that was like last week, September 1, right? We met last week. And then we actually detach the cell, detach cells. And then we transfer cells on 96 wells. And then what we have done, day 2, we treated chemicals. What are the chemicals? One chemical is cisplatin, the other chemical is PT-DECA2, right? So day 2, we treated. Treatment option will be here. So we have a little bit of a change here. If you're looking at it, this is columns, column 2, 3, and 4 of 96 well. And then we have a row A through H. So if you're looking at row A, we have a DMEM control. We call this as NTC. NTC means non-treated control. So we have cells in here. How many cells? 10,000 cells. And then we treat it with a media, which is DMEM, but nothing else. So we call it non-treated cells. If you're looking at your lab handout, I wrote down clear details about what these 3 wells means, okay? 10,000 cells and DMEM alone. And then if you're looking at row B, we have DMEM plus DMSO. So we call this row as DMEM plus 2% DMSO. So a lot of people asking, okay, I understand DMEM because DMEM is culture media. Why are we treating DMSO? Well, I have to tell you. See, cisplatin, listen carefully, cisplatin and PT-DECA2, they're not soluble well. They're not well soluble in water. So you have to solubilize these 2 chemicals in organic solvent. So one of the best well-known organic solvent is DMSO, right? So this row is another control that has media and a little bit of DMSO. So this is second control, right? So far, I hope that you understand why we're treating DMSO. And then now look at, this is a little bit confusing, but I want to get really clear on this. You see chemical 1 is cisplatin. So we're not treating a fixed amount of cisplatin, but we treated a varying amount of cisplatin. A little bit low, medium, and large amount. So let's see minimum or large amount of a chemical one, which is cisplatin. Look at this. These three here is chemical one, which is cisplatin 1 millimole, 1 millimole, 1 millimole. You may say, why you do 3 times? Well, we do triplicate, in order to increase accuracy, right? That's why we're treating 3 times. So same stuff, repeating 3 times. And then medium concentration would be 0.1, 0.1, 0.1. You see that, right? And then third, we have lowest concentration of cisplatin, 0.01, 0.01, 0.01, right? So those are group 1 and 2 and 3. And then now we have to treat chemical 2, which is PT-DECA2, 1 millimole, 1 millimole, 1 millimole, triplicate, 0.1, 0.1, 0.1, triplicate, and then 0.01, triplicate. So let me see whether I miss anything from this slide. Why DMEM alone? This is non-treated control, we said that, media alone. And why DMEM plus 2% DMSO? Because DMSO is able to solubilize these chemicals. That's what I covered. Why differing amount? You never know whether low amount will work better or high amount will work better. You don't know. That's why we're given 3 different concentration gradient here, right? In our case, triplicate. And what here we see 1.1, 0.01. While we're making this, we do serial dilution. The reason why we do serial dilution is increase accuracy. Another way we say decrease error, right? Errors. That's why we triplicate and we do serial dilution, decrease error. Okay, so these are day 2. Day 3. So if you're treating chemicals, what you have to do in day 3? Measure cell proliferation. Another name would be what I say, viability. Okay, viability or cell proliferation. Day 3. So in day 3, we do XTT assay. This is one of the best well-known. I think you might have heard of MTT assay. It's very similar, but we're using XTT, one of the latest assay that will be basically allowing us to measure viability or proliferation levels of cancer cells. So for this assay, we're using 2 chemicals, okay? 2 chemicals. One is XTT salt, and then second, we have a PMS. So all those mechanics of how XTT and PMS works will be coming next 2 slides. So basically now you know that for this assay, require you XTT salt and then PMS, right? So basically you will see, so day 3, what we do, we're treating XTT salt and then PMS together in a specific ratio and incubate about 5 hours. Okay, the reason why I underline here is because we didn't do any varying amount of time, but fixed time, 5 hours of a treatment of XTT along with PMS in the cell culture. And over time, this 5-hour period, the XTT will be reduced, right? XTT will be reduced, and then color change is associated. What color? It's gonna be orange. So the darker the orange, the more cells are viable, right? So I wanna say one more time. the darker the orange, the higher the viability, or the higher the proliferation. Does it make sense? Yeah. So let's see. 2 slides about how XTT and PMS works. This is XTT. The full name, never even trying to pronounce it. It's because so long. For the class, XTT is sufficient. It has number of benzene rings, right? And once you have a lot of cells viable, or they are proliferating rapidly, actively, then this XTT is reduced and become formazan derivatives, which is orange. This reduction, right, process is actually assisted by PMS. If you're looking at here, this is XTT. It gets reduced, orange. Now look at PMS. RED means reduced. Look at this. Reduction of XTT is coupled with oxidation of PMS. So I will say this, XTT reduced. This is very important. And PMS is oxidating or oxidized. Okay, make sure this. So you may say then, okay, PMS is helping reduction of these XTT. So then how PMS is coming back its original status? Well, it's because of our reducing powers. You say NADH and NADPH are reducing power. These NADH and NADPH, I think you already may be thinking of that, is created by glycolysis and the TCA cycle. Right? Glycolysis happens in the cytosol, and TCA cycle happens in mitochondria. I think you know this. So if cells are very active in terms of metabolizing, then they will produce NADH. If cells are active in living, then they use mitochondria, produce NADPH and NADH. So the higher the NADH and NADPH, it will represent the viability of the cell. If you have a higher amount of NADH and NADPH, well, you get more XTT. Does it make sense? One more time. If your mitochondria and cytoplasm metabolisms are occurring normally and actively, then you have a more XTT orange final product will be created. That's why I'm saying previous slide, the darker the orange, the higher the viability of the cell, okay? I probably told too many times redundancy. One of the bad things about teachers is redundancy. So here the kind of a hypothesis, null hypothesis would be this. There is no difference between any of the groups. What I mean by that, we have 8 groups of treatment. NTC, and then DMEM plus 2% DMSO, and then 1 millimole cisplatin, 0.1 millimole cisplatin, 0.01 millimole cisplatin, and then 1 PT deco, 0.1 PT deco, 0.01 PT deco. So we have 8 groups, right? So question is, null hypothesis, there is no difference between any of the group. Well, whether you're treating cisplatin or platin, deco, there is no difference. Means there is no effect on cancer cells. That's not hypothesis. Alternative would be this, or maybe 1 millimole high concentration of cisplatin and maybe PT-DECO will have a biggest reduction on cell viability. That would be a kind of alternative. You could set up different alternative hypotheses, okay? But this is one of the alternative. Once you set up alternative hypothesis and null hypothesis, what you want to do? Well, you got to test that. In order to test, you got to do experiment, as I already explained it, right? And so after we done all experiment, we get this kind of data. If you see that, this is DMEM alone. And then this is DMEM plus what? 2% DMSO. And this is 1 millimole cisplatin, 0.1 millimole cisplatin, 0.01 millimole cisplatin, 1 millimole platinum DECO, 0.1 millimole platinum DECO, 0.01 millimole platinum DECO. If you get any questions so far, please let me know. Experimental setups. We have 8, 1, 2, 3, 4, 5, 6, 7, 8 groups, right? So let's say from now on, you need to help me out. Everybody, you have I hope that you have computer. Well, let me see. Because you're looking at your monitor with a relatively bigger, you probably maximize this monitor so far, right? I don't know whether you can use Excel as of now. Let me know. Sophia, your mic is open. So do you think you can use Excel?
Yeah.
Okay. So how about some others? Let me ask questions to other students. So we have a 24 student, maybe. So I wonder whether you guys can utilize Excel. If
Yes.
you can, please raise your, you could use some kind of a, participated in showing your hands whether you can do Excel. Okay, good, good. Yeah, if you cannot use your Excel, that's fine. But if you can, I think it's better to use it, right? Okay, now, so if you open your Excel from now on, and you wanna type in Excel sheet these numbers, okay? So let me minimize this, yeah, I think this one. And show, at the end show, okay, and show. I wanna keep it. So you can open your Excel. Where is my Excel? It's here. So try to type these numbers like this, okay? I'm gonna stop the video. More, pause your recording. Awesome. So in order to add your standard deviation values, let's say I'm making it a little bit smaller, like this, okay? So, and make this one bigger, like this. So you still click here, chart element, but before you click chart element, you have to click the bar, like this. Click bar first, bar, or click the bar, one of the bar, then it will select all things. And then you go positive, right? Chart element. And then have to move a little bit. So here, you see that you want to go error bars, okay? Go error bars, and click that. One more time. So go positive, error bars. You don't say yes because all error bars are same. That's not correct one. So you go here and you go more option, okay? More option. So let me click it. Now you get this. Oh, wonderful. So, well, I want to go both top and bottom. And then you don't do fixed value. You're not going to do fixed value. You're going to do custom value, okay? Custom. Custom and specify values. And then what I will do, positive error value will be shaded. First, erase this value and shade like this. Make it a little smaller. So this, not this one, this one. Standard deviation values. You're supposed to have 8 of them. 1, 2, 3, 4, 5, 6, 7. Okay, that. And then negative error values, also the bottom one. And then, okay. So now let's see the data. You see that standard deviation window or values are varying. It's not all identical, but different. This is real data. This is how not only molecular biologists, but also ecologists, when they want to show standard deviation, they do this way. Okay. Okay. So am I sharing? So can you see my monitor? Min? Okay. Sounds good. Sounds good. Okay. In this way, you do this. Now, let's go back here. We have done pretty much here. Min, can you see this PowerPoint? Okay. Usually,
are there going to be instructions on like building that graph? Because I got a little bit lost when you were doing it.
The best way is this. You know how to make an average bar graph, right?
Yeah.
Okay. In general,
I got lost with the error bars.
Okay. So, okay. Let me see. So let me say, so what I could do this is, you see this is standard error bar, right? I can remove it. So you know how to do this much, and how to do standard error you want to add. So you click the bar first, okay, like this. Did you see that?
Yes, I did.
Okay. And then you see plus here, right?
Yes.
All right. And then you click it. You go down to error bars. You follow me?
Yeah, mine's a little bit different, but I'm following.
Okay. So maybe you're using Mac or something, it's fine. You can also, this video is recorded, so you can see one more time. So don't click it yet, but here you see arrow here, right? Arrowhead here.
Yes.
You click arrowhead and then go more options.
Okay.
And then you see this kind of stuff and choose custom. Custom. And it'll allow you to specify your values. So you hit specify value, and then it'll show up like this. Custom bar errors, question mark, and you have to select positive error value and negative error value, like this.
Okay.
Right? So what you can do, well, still this video is recording, so you remove it, remove, and bottom one also removed. So make it blank. And then you go back to your data for the here positive error value and shade it. Let me make this graph a little bit. I cannot do that. Okay. So I have to shade these values 3, 4, 5, 6. Standard deviation values. Okay. 5, 6, 7, 8. And then also negative error value, also copy the same data. Yep. And then you hit OK. You see that? So
Yeah.
because the video is recorded, you follow step by step, you should be able to do this.
Okay. I think it's because I'm on a Mac, so the layout's a little
Okay.
bit different.
Oh yeah, I have a Mac in my house, always different. I understand that. Okay,
Thank you.
thank you for asking one more time so that I was able to repeat this. So, once you get this kind of graph, then, I'm interested in, basically do statistical analysis using Excel. So what I usually do, uh, I usually go with these data here. What is that? Sheet 1, sheet 2. Yeah, here. Let me combine these guys all. Delete, delete. So these are raw data, right? Raw data. And I know you made a graph like this, but okay. So how can find out whether we have any differences between groups, or we have any differences within the group, right? How you know that? It looks like the numbers are very similar, but some of them 0.4, some of them 0.2, look like a little bit different, but your eye does not give you statistical data. We have to rely on Excel, software package here. So if you're looking at your Excel data chart, you may be able to see when you click data on your Excel, here, right there, data. You may be able to find out data analysis. If you don't have this data analysis icon, you might want to go to install the tool pack for this data analysis stuff. How you do this when you have a Mac or when you have a window, you follow this. You go tools, let me follow. You go tools. Of course, you have to go what is that? Options, okay? I think when you go Excel and options, you should have this kind of options. And then you may have, where's the tools? This maybe not. Let's follow this way. Tools and Excel add-ins, file option, okay. File option. And then you go add in here. And then you go analysis tool pack. Well, yes, in the second row here, analysis tool pack. If you click it and okay, now you're gonna get this. Data analysis, okay? Well, right now we are recording, so you should be able to follow this one at a time after class done, and you should be following me the way I'm doing. So, as I said, let me continue. By looking at the numbers, you don't know whether they're statistically different between groups and within group. So how are you going to find out differences? How are you going to test whether null hypothesis is correct? Null hypothesis states that there is no difference between groups, because whether you're treating cisplatin or platinum deco, there's no difference. That's null hypothesis, right? How you test that? Well, we rely on statistics. How we do this is like that. Click data analysis, and you get ANOVA single factor. So top one, you choose ANOVA single factor, and you say, okay, wonderful. And then, okay, what are the input range? Well, input range, I shade all data point like this. So you click here first, and then go there, shade it. That's your input range. And then you keep, alpha 0.05. This is value. And then now you have to click output range, output range. So you just click here, output range, and then you hit inside here a little bit, and then you click anywhere here, probably like this. Click, and then okay. Now you get the statistical data showing up. If you cannot follow me, that's fine. But all the specific details, the video is able to record all these, so you should be able to do this. Look at this, 8 different treatment, right? And then you see one of the most important things is this. In this Excel ANOVA single factor test, you have to look at F value and F critical value. These two values are actually one of the most important values that we really care. If you're looking at F value or F statistics value, if F value is higher, way higher than F critical value, in this case, 34 versus 2.6. So F value is way higher than F critical value. Then what you can say that is, oh, there is differences between groups, right? In terms of average values. That means what? Let me go back to our PowerPoint. Here, right there. If your F value, in this case 34, is larger than your F critical value, then you should reject the null hypothesis because you see the differences between groups in terms of their number. You cannot say that treatment of cisplatin will not affect any cell viability. It really affected. So you have to reject null hypothesis. Then you have to set up an alternative hypothesis. Alternative hypothesis would be that, okay, treatment of platinum deco or cisplatin will give rise to some differences in terms of cell viability or proliferation. Okay? This single ANOVA test will tell you, oh, there is a difference between groups, but they will not tell which one is showing differences. So we need to go another statistical package or software to figure out p-values, student t-test p-values, right? So probability of the differences between groups, So we're gonna go now, Prism 8, okay, Prism 8. So I click that. I don't know how many of you were able to download this Prism 8 toolpack. It's free for the next 30 days. If you have not downloaded yet, no worry, but at the class, you wanna download and then do as I do right now, okay? Okay, so when you download Prism 8, it'll go 30 days free. And then as soon as you open, you will see like this, data table, whether you're gonna choose enter or import data into a new table or start with the sample data to follow a tutorial. Don't click the bottom, but top one, enter or import data into a new table. And then x-axis is number, you choose numbers. And then y-axis, you click enter here, where enter 3 replicate values in side-by-side subcolumns. Remember, side-by-side, not end-to-end, but side-by-side, okay? And obviously you can click it, you create it. So now you get this kind of a group A, group B, group C, group D, group E, like that. And then title, right? So that's group title. How many groups do we have? 8 groups. So I type here, for example, NTC, non-treated control. And then here, DMEM plus 2% DMSO. Good. And then 1 millimole cis, right? 1 millimole cis. And then 0.1 millimole cis. Wonderful. It's going well so far. And then here, 0.01 millimole cisplatin. Good. Let's move. Group F. Now 1 millimole deco, cisplatin deco. And then here, 0.1 millimole deco. So you give all title. 0.01 millimole deco. Then you basically type in your value here. So let me see the value one more time. The value was here. So the way that I can see both would be like that. And you move this one down like this so I can see both. So I go group A, NTC. So X, so you see that this is data 1, data 2, and data 3 of your NTC. So you type in here 0.405 and side by side 0.397, 0.409. Right? You write this. And then I go continue DMEM, so 0.376. Oops. You can often make a mistake. You try to go down a little bit, but side by side. 0.371. Oops. And then 0.352. And then 1 millimole cis. 0.247. Min, can you help me the numbers so that I can focus on typing? What is the second one? Unmute, please.
Okay. So
now group D.
Group D.
Point.
So it's 1 millimole size, right?
Yeah, I'm
so,
writing already.
The first one is 0.247.
No, I think 0.1 millimole. 0.1 millimole cis point. So 0.367 and 0.368.
0.377.
377. Next.
0.396. 0.397.
Next.
0.4.
Next.
0.22. 0.232. 0.239.
Next.
0.463.
Oh, 63. Next.
Yeah. 0.364.
624.
Yeah, 64.
0.364. Next.
0.382.
82. Next.
0.394.
Next.
0.399.
0. Okay.
Yeah. 0.402.
Okay. So you typing this way, side by side. Oops. Okay. Oh, the cursor is moving so fast. Something is weird. Hey, cursor, stay there. Ha, I see that. Okay, now it's stabilized. So let's go back to the group A, like this. So we have all the data here. Now what we're gonna do, we go to analyze. I don't think you can see very well here. There is an analyze here right there. I don't know how many. Can you see this? Analyze. Analyze. Okay. So I hit this. And then, as I shown in your lab handout, you have to choose a specific button here in this case, one-way column analysis. We have to click column analysis. And then you choose one-way ANOVA. One-way ANOVA. Then you have 8 groups here, NTC and 8 groups. And then what you have to do, you have to go yes. Okay. So then we have this kind of a diagram, which is also given to your lab handout. And then we choose yes, use ANOVA. And then you choose yes, we use ordinary ANOVA test. And then you basically hit OK. Hit OK. Right. Oh, actually, let me go back. Data 1. Analyze. Choose column analytics. One-way ANOVA. Yes. Ooh, we have already performed. Okay. Here, before you hit OK, I think we have to go some other things too. Most cases, student and myself making mistake, just hit OK, and then later we have a problems. Okay, so here, what we have to do here, we have to go multiple comparisons because our goal is getting p-values. So comparing NTC with the, DMEM plus 2% DMSO. Compare NTC with 1 millimole cisplatin, something like that. So every individual comparison between NTC and other components. So if you click multiple comparison, then you choose compare the mean of each column with the mean of a control column, right? That's very important. And then what we could do, we could go option, under option, click it. You have to make sure that we're using a done it test. So you guys, you guys preparing exam 1 that will be ready for September 16, you want to know what kind of statistical, what kind of a multiple comparison test you use. If you have this kind of question, you should be able to answer done it test. Okay, done it. And then you click report multiplicity adjusted p-values. This is a probability values for each comparison. And what else? You're gonna do this and show this many significant digits, like 4 digits. Okay? And then finally you're okay. Now I'm on okay. Boom. Now we have ANOVA results. You see F values here. F value is 34. Don't you remember when we did Excel, we get F value of 34, right? 34. Now we get, yes, F value is 34. That means what? Whether you're using Donut statistical package or we're using Excel single ANOVA test, F values are way higher. F critical value. Right? F value is way higher than F critical values. So you have to reject null hypothesis. That's the main point. But our goal was using this, Prism 8 statistical package is to get multiple comparison. Whereas actually statistical significance exists between groups. Well, you see this. Look at this. P-values. So p-values are here, adjusted p-values. When you compare NTC with the DMEM plus 2% DMSO, well, statistically not significant. There is no difference. How about NTC with 1 millimole cis? Oh yeah, they're statistically different. P-values lower than 0.00001. If you do 10,000 experiments, one error will be occurring in terms of, experimental error. But otherwise, you always get they're different, okay? And then NTC versus 0.1 millimole, statistically not significant, NTC 1 millimole deco, yes, statistically different. P-value lower than 0.0001. If you do 1,000 experiment, 99 9,999 times, you get similar data, always different. But if you go lower concentration of PT decals, they are statistically not significant. So you have all p-values right there. You have also statistical significant or not significant written here. NS means not significant. And you see asterisks, they're all significant. Right? You have 4 zeros, 4 asterisks, 4 zeros, 4 asterisks. So you now know that, oh, so we did very well to rejecting null hypothesis because we got good data here. Okay. Now what we have to do. So we know p-values. So what we have to do, we have to make a graph. To do so, we have to go data one. If you have not renamed, so graphs. We have to find the graphs. Where is the graphs? You're right there, graphs. Right, graphs. And I hit data one. So it will show this. This image is copied and pasted on your lab handout, so you should be able to see that. So you click in summary data, grouped. So grouped summary data and mean with the standard deviation. Right? So that's how you're going to do this. So it's pretty much default. And then simply what you have to do is okay. So you get this data, but I think they're very heavy. So I'm going to actually move all these guys. Oh, I don't know whether I can move these all guys. Hopefully you can move it. Let's say, oh, they're moving, wonderful. Let's move it, move. So legends are too close, so I move it. And then these guys, I want to extend it to see the bottom very well. So extend it like that. Let's go a little bit more. And maybe this one extend a little bit. And that one. So like this. So you have a, so 8. So you can title data here, XDT data. And then Y, X title will be treatment, right? Treatments, like that. And then Y title will be something like cell viability / proliferation. You can type this way and you enter it. Oh, enter. Oops. Oh, you hit somewhere else. So then you go cell viability and proliferation. What you can see is this: the higher the bar, the more cell viability. The higher the bar, the darker the orange. You see the NTC is higher, and lower concentration of cisplatin, the bar height is equivalent to that of non-treated control. But only if you're looking at 1 millimole cisplatin and 1 millimole decal, they show lower levels of a cell viability. That means pale orange. It becomes orange in terms of color change, but not darker. So that's what you can see. But it's not done because this 1 millimole cis and then 1 millimole decal, they show p-value extremely low, right? So you know the p-values. So what we can indicate p-values by go here, draw here, okay, draw. So you click here, draw. You could use here. This kind of bracket, right above the bracket, you can indicate the p-values. So I would choose this one, that, and then you see the pencil. So you compare NTC along with this 1 millimole cisplatin. And they show 4 asterisks, right, before, so you choose 4, like that, wonderful. And then between NTC and 1 millimole decal, you're gonna add one more comparison, and then you choose 4 asterisks. So this is a completed version of your data, right? So when you're writing your report, you need to include this data. So that means you need to download Prism 8 and watch this video, move step by step as I do, and should be able to show this bracket with the p-value indicated by 4 asterisks, showing average bar plus standard deviation. Here they creating standard deviation automatically. So you don't need to do too much worry about it. So this is your data, right? And then if you're looking at your lab handout, I'm bringing here, pretty much everything is listed here. And then if you go around practical questions, and I ask a few questions, like particularly number 8, if you're looking at it, use Excel to figure out those values. You can provide your Excel table here. Instead of you writing every numbers, you can copy, paste here in order to answer correctly, right? Or if you wish to go back to your Prism data, data 1, not this one, here, not this, that one. If you're looking at that, you have all standard here error values, mean values. Yeah, oh, actually mean here, mean 1. So mean values here, I think this is standard deviation. But you can go, Excel and you can copy paste that. And you know F values and P values and F critical values. And P values, you may say, where's the P values? Well, if you go back to Excel, you should be able to see P values right there. You're writing this 1.662 to the power of negative 08. So basically, so all data values are given. So you should be able to number 8, number 9, and number 10, you're providing your data here. Process data with Prism, not with the Excel, right? So you're providing this. And p-values will be asterisks, or you can type these p-values right here. Lower than 0.0001, lower than 0.0001, like that. You can type it, right? So, and then if you're looking at some questions here, you should be able to answer this, the relationship between levels of color change and cell viability. I probably repeated multiple times. And I also talk about what PMS does for XTT assay. And then chemistry of XTT reduction. I told you that some kind of mitochondria is able to particularly focus on mitochondria. Mitochondria is able to produce NADH, NADPH. They are reducing powers. When you have a high levels of these reducing powers, and then PMS will be activated and cause reduction of XTT, right? We talk about that. So you're gonna summarize that here. When you're using platinum-associated chemotherapeutic agent, you gotta be very much follow the policy, particularly dealing with the toxins, because it has side effects, particularly fast-growing cells, like your fingers, fingernail, and your hairs. They are really fast-growing cells. So if you mishandling these toxins, and what happened? You're losing your hairs will die, and you lose your fingernails like that. So that's why you got to be extremely careful. And then number 3 will be a little bit tough question: how to make 50 millimole cisplatin, molecular weight is 300, in 100 milli microliter. Okay, so you have to convert this one particularly. 100 microliter is 0.1 milliliter. 0.1 milliliter. And then you have to convert 0.1 milliliter into liter. So 0.1 milliliter is like I don't know how many zeros. If you have a calculator, you should be able to do this conversion. And then you can do the way I explained while I was explaining how to making molar solution, okay, molar solution. And then hypothesis, you should be able to set up because we practiced it. And the action mechanism of cisplatin should be found in earlier pages of lab handout, okay? And finally for this class, exam will be coming next week. So I wanted to give you a little bit of a study guide. Of course, I will post this on the Blackboard, but I want to give you a quick guideline of how you're going to prepare your lab questions for the exam 1. All exam will be multiple choices. As far as I know, 10 questions from the lab. And Dr. Durham will be telling you about lecture exams. So I'm not going to tell you more details about your lecture exam. For the lab, you should be able to be very familiar with these type of questions, like this. How would you make 250 ml of 2 mole NaCl solution? Of course, multiple choice, we're not going to ask you to show your work, but you will have to find out answer from multiple choices. How would you make 30 ml of 35% sucrose solution? Make 30 microliter of 1X restriction enzyme buffer from 20X stop. Make 20 ml of 0.5% of NaCl from 10% stop. These questions are highly related to the problems, problem set that you solved before. The numbers are a little bit modified here. An exam will not give you exact same number. So if you just memorize these numbers, you're not going to get any correct answer. Okay. And this will be posted. And then lab 1 unit change. For example, how many milliliters make 0.2 liter? So you should be able to practice it. Get familiar with the conversion questions, milligram versus gram, microgram versus milligram. And then you should be able to know the role of XTT assay. Why are we using XTT assay? What was the purpose? Yeah. And if you know XTT assay, the purpose, you have to know action mechanisms of XTT. So we'll go telling about that a little bit later. What was the role of PBS during lab 2? What was the role of trypsin EDTA? What was the role of FBS, fetal bovine serum, after treatment of trypsin? Right? So please study that. And how you counting using hemocytometer. So if you have whatever number of cell, average number of cell, how you want to convert that, the numbers seen on hemocytometer to be the real number. So practice that. And then how XTT and PMS works. I already covered this. And then why are you using DMSO in our experiment? Look at your, PowerPoint slide or my explanation, my video. And how you interpreting your PRISM data. How you want to interpret that. So those are kind of a quick guide for exam 1. Any questions so far? Sorry that I had to talk too much myself. Anybody, any questions? Welcome. If you wish to turn on your video, I'll be appreciating. Some of you, if you are able to turn on your video. Okay, thank you. Bryce or Betty? Your first name is Bryce?
Yes, that's my first name.
All right. So I saw you last week, but I cannot match your name and your face. Brittany, thank you opening your window. Any questions so far? Andrew. Okay, so first of all, what I'm gonna do, pause record.